Where's the other one? Okay, we've been recording the whole time. That's fine. We just need the YouTube thing. There we go. Right. So, hello everyone, and welcome to session 30. Echo. Fantastic. Hello everyone, and welcome to session 36 of BGA24. This session is on ENA Submissions and Data Standard by Myra. Please take it away.
Thank you, Damon. Hi, everyone. My name is Myra. I'm a user support bioinformatician at the European Nucleotide Archive. Just before I start, I am working with a small screen today, so if at any point there's something wrong with my display or how I'm sharing my screen, please let me know and I'll try and fix that. I will just start sharing my screen now. Hopefully everyone can see that. If not, let me know. Okay, great. So today I'm going to give you an introduction to the European Nucleotide Archive. I'm going to talk about the data standards that we have, how we structure our data, and the data submission routes that we have at the ENA. So I will start with a background of the ENA, what the European nucleotide is, what kind of data we accept, what kind of collaborations we have, why you should be submitting data to the ENA, and the kind of usefulness, the reasons people do submit data to the ENA, the data standards that we have. So we structure our data using two main models. We have the data model and the metadata model. And then we'll dive into data submission at the ENA, and I'll have a Q&A session, and then we can move on to a short practice session where you will have a chance to submit some data yourself. Right, so what is the ENA? The European Nucleotide Archive is a repository of the world's nucleotide data. Essentially, we accept all kinds of nucleotide data, whether it is DNA or RNA, whether it comes from plants, humans, or animals, as long as it is not restricted access, identifiable data, it comes into the ENA. We are an open access archive, so if you do have data that is subject to GDPR or some kind of private data policies, then it does not go into the European Nucleotide Archive. But if this is open access data, it will come to us. We have a whole range of tools that can assist in this kind of data submission and data retrieval, depending on the volume of data that you're submitting, the type of data you're submitting as well. And we'll touch upon this in just a moment. We are the European node of the INSDC. This is the International Nucleotide Sequence Database Collaboration. And all this essentially means is this collaboration is formed of the ENA here in Europe, NCBI or SRA or GenBank in the USA, and DDBJ, the DNA Data Bank of Japan in Japan. These three repositories mirror and exchange data on a daily basis. So if you submit data to one of us, it will be accessible on all three archives. So you do not have to submit data separately to the ENA and separately to GenBank. As long as you are submitting data to one of us, it is exchanged between all three archives and it is accessible more globally. Right, so why should you submit data to the ENA? And we know that all data submitted to the ENA is submitted by members of the research community, but what motivates them to submit this data? And there's a list of reasons here, such as open data, data reproducibility, reusability, so to keep it in line with fair data principles, any kind of third-party access, or ownership of data. So when you submit data to the ENA, you are the owner of that data and your name and your institute is affiliated with that submitted data. Archival of data as well, so you don't have to worry about locally storing large volumes of data. Once you have submitted this to the ENA, you don't have to worry about preserving this data, making sure its metadata is kept along with this data. Once you've submitted it to the ENA, you don't have to worry about that. Publication, of course, so majority of reviewers these days require that your data be available in an open access archive. And that's where the ENA comes in. So this data would then be submitted to the ENA, and the project accession would be cited in your publication. You can also link your publication to your data on the ENA, and we will have a look at this in just a moment as well. Also, any downstream analyses that you want, and Magnify right here is a good example. I don't know if you've heard from Magnify in this course, but Magnify offers this kind of additional assembly and analyses on metagenomic data that has been submitted to the European Nucleotide Archive. Right, and this diagram right here just shows how the ENA is linked to a whole range of EBI resources. And this kind of integration into these different resources really contributes to the value of its genomics data as well. As a few examples, you have Ensemble, which acquires assembly data from the ENA, and they provide annotations to this data. You have UniPro, which pulls CDS sequences from the ENA, and they translate these into protein sequences. AirExpress, which stores data from high throughput functional experiments, and any raw data that is associated to this data is stored at the ENA, and it's linked. And you have Magnify as well. So you have a whole range of EBI resources that are in one way or another linked to different data objects within the ENA. The ENA is also recognized as an Elixir core data resource and a global core biodata resource by the GBC. Right, moving on, how do we structure data at the ENA? So we have two main models that we use to present data at the ENA. This is the data model and the metadata model. And the reason we have this is because we're storing a huge volume of data, and this comes from many different researchers from many different places who work with a whole range of different species. They use a range of different techniques, and the sequencing is done on different platforms. And what we need to be able to display this in a consistent manner so that anyone who's trying to retrieve this data can easily be able to do so. And the first model that we have for this is the data model. And this is what the data model looks like. So we have three main categories. You have your reads, your assemblies, and your annotations. Your reads is just the raw output of the sequencing machines. So this category is when very minimal to no kind of analyses have been done on this data. The most you have done is maybe demultiplexing the data, but that's about it. So this is your raw read data. You then have your assemblies, in which you've used some kind of algorithm or software to assemble these sequences into a genome or part of a genome, and that counts in the second category as genome assemblies. And then you have your annotations, which are interpretations of biological functions of these sequences. And often you can group assemblies and annotations together, and you can submit these as annotated assemblies. Right, so these are our three main categories of data that we accept. We also accept other types of analysis data, but broadly speaking, these are the three main data types that go into the ENA. Now, the second model that we have is our metadata model. And this figure right here shows what the metadata model looks like. And the reason we have a metadata model is to provide a whole range of metadata values and fields according to the type of data that you're submitting. And I quite like this example. So if I want to see bacterial data taken from the Atlantic Ocean, sampled between a specific depth, between a specific time of year, compared with exactly the same from the Indian Ocean, I can do that very easily because data has been submitted in accordance with this metadata model. And this is what the metadata model essentially is. It comprises of a study object right here. This is kind of the head of your project. So this is what all data that you submit will be grouped under the study object. So this describes what this project will contain. You then have your sample object, and this contains metadata in relation to the biomaterial that you have sequenced. You have your experiment object. This contains metadata concerning the sequencing, the library. You then have your run object and your analysis object, and these are these two blue objects are essentially the only ones that hold your data files. Everything else holds metadata associated with these data files. Your run object right here stores data that comes from the first category, which is our raw read tier, and your analysis object can hold assemblies and annotations, any kind of downstream analyses. And just to explain this in a little more detail, I'm going to give you an example. So this is an example of viruses in bees. So viruses have a range of effects in bees, and this was studied by one group where they sequenced viral metagenomes from 11 bee species from 9 countries. They also developed a pipeline to assemble these into contigs. So this right here, this description of what the project looks like is our study metadata. And this entirely goes under our study object right here. So reading this, whoever is trying to retrieve this data would have an understanding of what type of data would be included in this project. And this is how you register a study at the ENA. So it's essentially a web form that you fill out, and you fill in details like a study name. You give a title to your study, you give an abstract, so like a paragraph describing the study. You can also link any publications for this study, and if you essentially don't have any publications while you're registering the study, you can always come back at a later date and you can add your publications later on. And this right here is a release date, and I like to focus on this a little bit more just because a lot of people don't know this, but when you submit data to the ENA, it does not mean that your data will become accessible to the public right away. You actually have the power to keep this private for as long as you want. And this release date you can set to 2 years from today, and you can keep postponing this release date. And when you are ready to release your data and when your publication's ready, you can come back here and you can prepone this release date to tomorrow or next week or as soon as you want. This is really useful because essentially when your publication is ready and when a reviewer asks you to submit data to an open access archive, that's when a lot of researchers start submitting their data, and that's when it's a bit chaotic, you're in a bit of a rush, and you run into, your data files can essentially run into errors, validation errors that we then send to you, and then you have to try and get in touch with us and figure out what's going on, what's wrong with the data files. This way, you have your study, you can register it, you have your raw reads sequenced, you can submit them. You don't have to submit your assembly data yet. Your data is private. You can set your release date to as far as 2 years from now. You have your raw reads, you submit them, and you don't have to worry about keeping them preserved. You then have your assembly data or your annotation data. You can then come back and you can submit those under the project as well. And all of these would have been processed by the time that you are ready to release this data. So you essentially won't run into many problems. Right, and that is our study object. Moving on to our sample object, which comes next. In this example, collaborators from 4 continents sampled these bees, and they recorded some sample metadata fields like the GPS coordinates, the species of bees, and they had a total of 37 separate samples. Now, this is the kind of sample metadata that you would record in order to submit the samples to the ENA. And this is what this would look like. So you have one study and you have 37 samples here, but just for display purposes, I'm showing you 4 samples here, and you can see fields like the location and the species of bees. And this is how you register samples at the ENA. So you make use of these sample checklists, and we have a whole range of checklists that you can choose from. These really depend on the type of data that you're submitting and what species you're working with. This is a host-associated checklist, so because you're working with viral metagenome, they're using the host-associated checklist here, and the kind of metadata fields that you have to provide also vary a little bit between these different checklists, and the fields are relevant to the type of data you would likely have based on your sample. So here you have things like tax ID, a scientific name, and then you have things like collection date, the geographic location, the geographic coordinates where this data was collected. And so these are the kind of mandatory fields that you would provide with your samples. We also then have recommended fields and optional fields in a lot of our checklists. And these are fields that you do not have to provide, but if you do have the values for these fields, if you have recorded these attributes, we highly encourage that you do provide these, because the more metadata that you provide, the better quality your data is, the better annotated your samples are, the more findable it is, the more reproducible it is. So it's very recommended that before you actually start working on this, you actually you familiarize yourself with the types of metadata fields that you might need to record. And then if you do have metadata attributes which are not included in our mandatory fields, recommended fields, or optional fields, you can always add them as custom fields right here at the top, and these will be included in your template. So just to reiterate, the more metadata that you provide, the better quality your data is. Right, this is just to highlight that again, and this is an example of including a custom field, which is then added to your checklist as a custom field, and it will be included when your sample goes public in the browser. Right, moving on from samples to experiments. In this example, the viruses were isolated, their DNA RNA extracted, and following amplification, the material was sequenced on an Illumina MiSeq. Now you have 37 samples, and you sequence them to get 37 separate single experiments. And this is what that would look like at the ENA. So you have one study, you have your 4 samples that are grouped under the study, and then each sample has its own experiment object. Now, experimental metadata includes things like library layout, the sequencing machine, the platform that you used. And these metadata values are actually provided when you're submitting your data files. So you register your experiments and your reads at the same time, and I will show you that in just a minute. Moving on to runs. The result of these experiments, 37 experiments, gave us 37 single FASTQ files, and you compress these to gzipped or bzip2 format. You upload them to the database and you submit them to us. And then they go through our validation pipelines, and they're waiting to be made public. This is an example of what a FASTQ file looks like for raw reads. Right, and that is how it would look like. So each experiment that is grouped under the study then has its own run object, and the run object is the one that contains your raw read file. And this is how you submit experiments and runs to the ENA. So this is similar to samples. You have read submission checklists, and this is one for FASTQ files. We have one for Oxford Nanopore data as well. So it really depends on the file type that you have. And then these are the kinds of metadata values that you would provide. Experimental metadata such as the instrument model you use, the library source, the library selection, library strategy, library layout, the file name that you are submitting with this experiment, the sample that this should be linked to, and the study that this should be linked to. Right, and that completes most of our metadata model. The only thing we have left is our analysis object. And at this point, if you don't have assembly or annotation data yet, you can leave it at this as well. In this case, read data for one of these samples was ran through an assembly program, and it produced a set of contigs. This right here is a FASTA file, and this is how it would look like at the ENA. So you have one study. You have 4 samples associated with that study. Each sample has its own experiment object. Each experiment object has its own run object, and only one of the runs and one of the samples are then linked to an analysis object, which are your WGS contigs. So this is the kind of metadata that you would provide when submitting an analysis object. So you can see here the assembly type that you are submitting, the coverage, the molecule type, the file name, the study this belongs to, the sample, the run this belongs to. And that completes our metadata model. So you've got one study, you had 37 samples associated with that study, all grouped under that study. Each sample has its own experiment object with its own experimental metadata. Each experiment object has its own run that contains your raw read data files, and one of the runs and one of the samples then are linked to an analysis object, which is your assembly. Now, the linking is quite flexible. So if sometimes you have cases where one sample is ran through multiple lanes, which gives you multiple runs essentially. So you can link one sample to multiple experiments and multiple read files as well. So the linking is quite flexible here. Right, and that was our metadata model. So we started with our study, when we then moved on to our sample objects. We then moved on to our experiment objects, and then we moved on to our data files, starting with our raw reads, and then moving on to our assembly data. And this is how it looks like in the browser. You can see right here is your study accession or your project accession. This right here is the metadata that you provided with your project. So the abstract, you can also access the XML here to see all the project metadata linked to this project. This right here is a run data table. So you can see here you have your study accession, you have all your sample accessions that you have registered. We then have the experiment accessions, the run accessions, the tax ID and the scientific name, the species that this belongs to, which is pulled from the sample metadata. And this right here are the FASTQ files that have been generated. This is just a point on the tax ID that you use when submitting your data. So the scientific name or the species name that you use while submitting your data, this should be as accurate as possible. And this is a good example. So if you are submitting metagenomic data or environmental data, it's important to use, for example, human gut metagenome as opposed to Homo sapiens, or mouse gut metagenome as opposed to mus musculus. So it really depends on the data type that you have, but your taxonomy should be relevant to the data that you are submitting. The browser also has this kind of navigation bar right here, and this used to be just one box. We've now separated this into two, so we have a general box right here, and then we have a data box to highlight the data files submitted with this project. So you can see things like you can access almost everything linked to this project through the navigation box. You can access things like cross-references, any publications linked with this project, any related ENA records, all the read files submitted as part of this project, and all the analysis files submitted as part of this project. The specific project only has WGS sets, so you only have a WGS files field here. But if I access, for example, the related ENA records, I can see all the data objects that are linked to this, the assembly data and the run data as well. And if I try to access, for example, the cross-references or the publications, I get to see something like this. So if I access the publications, I can see a list of publications that are linked to this specific project. So it's quite nice to link your publications with the projects. And then I can also, with cross-references, I can see anywhere that and this has been used. For example, magnify any kind of downstream analysis that they've done on this. Those can be accessed from here as well. So this is quite useful that way. Right, moving on to data submission at the ENA. So we have a few routes that you can use to submit your data to the ENA. We have three main submission routes. We have the interactive submission route, the programmatic submission route, and web and CLI. The interactive submission route is the most user-friendly route, and someone who is new to submitting data to the ENA can make use of this quite well. This is essentially all done on a browser. The programmatic submission route makes use of XMLs or JSON files that you can draft to describe all the metadata values and your data files, and you can submit them using curl commands. And then we have web and CLI, which makes use of the command line. So the interactive route looks something like this, like I showed you. So you have these web browser forms that you fill out. And it's almost entirely done on the browser, so it's quite straightforward. The only drawback for this perhaps is if you're registering a large volume of data, it can be quite slow. For example, if I am submitting even 15 projects at one point, I would have to register each project one by one, and this would be very time consuming, as opposed to the programmatic route, for example, where I can include them all within one XML or JSON file. So the programmatic route can also be integrated within your scripts. So if you regularly submit large volumes of data, you can use this to automate a lot of the submission process. And this is an example of an XML file for samples. So you can see that the metadata values and attributes are quite similar to, or the same as what I showed you in the checklists earlier. For example, tax ID, scientific name, collection date, geographic location. So it's the attributes that you provide are similar, but the file is in XML format or in a JSON format. You also have to include a submission XML along with this, which contains an action. If you're adding a new sample, you would use something like add here, for example. And you can submit this using a curl command. This is an example of what the curl command would look like. You would include your web and username and your password. The files that you are submitting, in this case, a sample XML and always a submission XML. And you would then submit this to the ENA. And the last route that we have is web and CLI. This is a command line interface. This is also the only way you can submit assembly and annotation data to us. So if you are submitting these kind of analyses objects to us, you have to use Web and Cloud. You cannot use the programmatic route or the interactive route. Web and Cloud can only be used to submit data files. So you can submit your raw reads using Web and Cloud. You can submit your assemblies using Web and Cloud and your annotations using Web and Cloud. But if you're registering a study or if you're registering samples, you have to use either the programmatic route or the interactive route. The reason Web and Cloud is the only route you can submit assembly and annotation data is because it offers this pre-submission validation, which is really useful. So when you're using the Web and Cloud software, before it accepts your submission, it validates it for you. And if there are any kind of errors in there, it does not accept the submission. It goes back to you with an error report. It says, these are the kinds of errors that your file is encountering. Please fix them, try again. So you can then have a look at the files. You can see what kind of validation doesn't match the ENA validation. You can make any fixes that you want, and then you can come back and you can try and submit it again. It won't accept your submission until it has been validated successfully. Once it has been validated successfully, you don't have to worry about any post-submission errors that your files might encounter because you know it has already gone through this kind of validation step. With the other two routes, you submit your data files and they come to us and then they go through our validation pipelines. And if they encounter any errors, we then email you saying they've encountered validation errors, and it's generally a longer process. Whereas with Web and Cloud, you can have a look at the validation errors yourself, and you can fix them, and you can try submitting them. You can also test your submissions with Web and Cloud. So if you're not ready to submit your data files yet, but you just want to test whether everything's okay with the validation, you can use a test flag, and this will test your submission, and it won't submit it. And it will let you know if the validation was successful, and you can be rest assured that when you're ready to submit your file, you should be able to submit it successfully. With Web and Cloud, this is an example of a FASTQ file that I showed earlier as well. With Web and Cloud, you also have to submit a manifest file, which is a plain text file, and you include the same kind of metadata values that you would if you were submitting interactively or programmatically, such as the library strategy, library source, library layout, and then the name of your file. And you then submit these into the command line so they can run through the Web and Cloud program. Right. This slide kind of just summarizes everything that I've said. You can see right here, these are our submission tools that we have, or submission routes that we have. We have the interactive route, the programmatic route, and Web and Cloud. And then you have your metadata objects, which are taken from the metadata model. You have your projects and your samples and your taxon that comes from your samples. These can be submitted programmatically or interactively. You have your read objects, so your runs and your experiments, and these can be submitted interactively, programmatically, or using Web and Cloud. You have your assemblies and your annotations, and these can only be submitted using Web and Cloud. So any genomes or context, chromosomes, any scaffolds and coding sequences, you have to submit using the command line interface. We also have a bulk Web and Cloud tool, so if you are submitting a very large volume of reads or assemblies, you can make use of the bulk Web and Cloud tool. And then we have a couple of COVID-specific tools as well for data submission. This right here shows some of the portals that we have developed as part of different projects. You have your pathogens portal right here, which contains all pathogens data linked from the ENA. We also have the COVID-19 data portal, which, as the name suggests, is SARS-CoV-2 data. You have the early cause portal, which links data from the ENA linked to any early life stress. Right. just to summarize, the ENA stores nucleotide data, whether it's DNA or RNA, from a wide range of species. It is also part of the International Nucleotide Sequence Database collaboration. In order to present this kind of large amounts of diverse data, we have two data models. You have your data model and our metadata model. The reason we have these models is so you would submit data with a minimal mandatory metadata requirement, which makes your data more fair. We also have different tools that can be used to be able to submit this data, and the type of tool you use depends on the type of data you're submitting, the volume of data you're submitting, and your own personal preference as well. We have three main data submission routes, the interactive route, the programmatic route, and web and cloud. Right, that brings me to the end of my talk. I'm happy to take any questions at this point if there are any. Otherwise, we can move on to the practical.
There's one question so far. What kind of checks does the pre-submission validation do?
So it depends on the data type that you're submitting. And if you're submitting, for example, a FASTA file, the validation checks include things like the format of your FASTA file, the sequence headers, the def lines. If you're submitting flat files, which are annotated assemblies, for example, then the validation is a bit more extensive. It looks at all the qualifiers and all the lines in your files. Likewise for raw reads as well. It looks at things like, for example, if you're submitting paired reads, it looks at things like the read names and the pairing percentages, any duplications. So it depends on the file type that you're submitting, but the pre-submission validation is for raw reads at least, because you can submit raw reads through Web and CLI and through the other two routes as well. The pre-submission validation is exactly the same as the post-submission validation as well.
Okay, cool. There's another one on Oxford Nanopore. Does ENA take POD5 and FASTQ files?
So for Oxford Nanopore data, we have been discussing POD5 for a very long time, but we haven't implemented it, and we have no plans of implementing it anytime soon. So I don't have a timeline for you on this. We accept Oxford Nanopore data as FAST5 files, I think, which are very large files, I understand, but that's the only format we accept them at the moment.
Is it possible to update the information, e.g. the name of a species, after the data has been released? Is it updated on NCBI as well?
Yes, so just to mention that anything that is updated in the ENA will also be updated in NCBI, so they're always in sync. Likewise, anything that's updated at NCBI end, it's always mirrored into the ENA as well. For changing species name, this can always be changed, and it can be updated because we do have cases where species names have been changed in the past. We also have cases where you have informal taxonomies before publication for novel species, and then you need to change the species name after the publication has been approved. This can be done as well. So on sample level, you can update species names and tax IDs after submission.
Cool. If you are sequencing a genome, would the study be the genome and then the data file types added under data?
I'm not sure I understand that too well.
Can I ask the question? Do you mind?
Good.
Thanks, Mara. So if you were sequencing a genome and you were generating multiple different data files, so for example, long read sequencing, maybe some Hi-C, maybe some RNA-seq, those would all be data files. But then would the actual genome be the study? So creating that genome assembly, would that be the study?
That wouldn't be the study. The study would describe this entire thing, but you will be able to link the genome to the remaining data objects. But the
Okay.
study would not contain any data files itself.
Yes, yeah. Just be a description of what you were trying to achieve with the genome assembly, ultimately, presumably.
Yes.
Okay. Thank you.
Right.
You've got another 3 now. Must the reads always be in FASTQ or can they be in BAM?
They can be in BAM format or CRAM format as well.
Cool. With annotation, is it mandatory, and do you accept any kind of annotation, e.g. liftoff?
It's not mandatory. You can submit your data without annotation, and Ensemble does pull unannotated data from us, and they annotate it as well. So it's not necessary to have annotation with your data. Yes, we do accept most types of annotations.
Cool. And for the assembly, do organelles have to be in the same FASTA as the chromosomes? Do primary phased assemblies of the same organisms have to be submitted together? And do you perform quality checks on these?
So for metagenomic assemblies, we do have quality checks. And if you're submitting chromosome level assemblies, you would submit a chromosome list file along with your assembly, which would contain all the chromosomes that your assembly file contains. And this, once it becomes public, these will be individual chromosome sequences, but they would all be linked to one assembly. Does that make sense? I think that was all. Is there anything else?
Is the organelle included in the
So-
main assembly FASTA?
Yeah, so one of the fields in the chromosome list file is to specify the organelle as well.
Cool. Now there's typing happening. Thank you. Cool. Right. Okay. I think that's it for questions for now.
Great. So we'll just move on to a practical. This should be fairly simple. We're just going to, oops, right. So this is an interactive data submission practical, and you will be registering a study, registering a sample using a spreadsheet, and submitting a read file. So this will all be done interactively. So we are using the first submission route to practice this. And hopefully we don't run into too many issues, but I'm just going to share a link with everyone. And if you just open this, you should be able to access the document. Right, so this is the practical essentially, and as I will just talk a little bit and then I'll let you start this on your own. But as we've discussed, there are 3 routes of submission. We have the interactive, programmatic, and web and try. For this practical, we're going to use the interactive submission route. So we will be using the web and portal. This is a link to the web and portal. What we're going to be doing is we're going to be using the test web and portal. And you can see it has a little dev just after let me just share this screen actually. Right. Yeah, so as you can see, it has a little dev right here after www, and this is the test portal. Anything you submit to the test portal is wiped every 24 hours. So any data that you submit today, it will not go public, so do not worry. This is just for practice purposes and training purposes. The test portal is literally just there so you can test your submissions. Just make sure that you use the test portal for the duration of today's practical. The first thing I need you to do is if you could just go on this link right here, the workshop accounts list. And if you could open this file and just add your name against one of the Webin accounts, and you will use that Webin account for today's exercises. Right, so just make note of the Webin username. So that is the full username. That's Webin hyphen and the number. The password for all the accounts is the same, and it's listed right here at the top of the document. And you will be using this account for all the exercises today. If you just scroll down to the scenario page. And so for these exercises, you will be submitting a read data file that is derived from mouse gut metagenome, and you will be submitting this to the ENA test service. To begin, like we discussed, the first step is always to register your study. So the first thing you're going to do is you're going to register your study for this mouse gut metagenome sample read file that you have. And in order to register the study, you have the instructions right here under the study, and they're quite straightforward. The study is one of the more straightforward steps, so I'm just going to give you about 10 minutes to go through the first part and try and register your studies. Once you have successfully done this, you will see that step 7 at the end of the study exercise is to add the study accession into the accessions document. So you will just add your accession right here. For now, just start reading the study instructions and try and register your study. Let me know if you have any questions in the meantime.
Oh, sorry, there's just been another question. Is there a tutorial for the WebCLI?
So we do have our documentation, which explains the WebCLI software quite well. I will just send a link to that in the comments.
Oh, thank you.
All right. I can see that you have managed to register your studies. Great job. We'll move on. Anyone who's still working on the first part, that's fine. But we will move on to the samples part of this exercise. And unless you want me to give a demo of the study registration, I don't think I need to, but for sample and for read submission, I will give you a demo as well. So if you just scroll down to the next page, which is the sample. So your next step after your study registration is to register your sample. And this is going to provide users of the metadata linked to what you have sequenced essentially. So this is going to be slightly more complex. You're going to work with a specific checklist. In this case, since we're working with the mouse gut metagenome sample, we will be using the MIXES host-associated checklist, which was used in the example I showed you as well. So you can download this checklist template from the Webin portal as well. If you just start reading the sample section and just follow the instructions, and please shout out if you have any questions. I'm usually drowning in questions by now. So the fact that it's so quiet is a good sign, means you guys must be doing really well. Or I'm a great teacher, either one. Um, right. If most of you are done with the samples section, if you could move on to the run, which is the final exercise. Great work, guys. So this is the last part. You've registered your study, you have registered your sample, and now you have to submit your read data. Now, the first step, usually when you're submitting your own data in this, is to upload your read files, which are your FASTQ files or your any kind of data files. In this case, the upload part has been done for you for these accounts, just to make this a bit more seamless. And you would be submitting these files. So this is quite similar to the samples section, where you would use a read submission checklist to submit your FASTQ file. So if you just go through the instructions for read data, and please shout out if you have any questions at all.
Quite nice. You've got some quite comprehensive error messages and submitting a bad form. Very helpful.
Sometimes they're comprehensive, sometimes they're completely ambiguous. So
Oh, lovely.
can be either. Give this another 2 to 3 minutes and then I'll do a demo. And we'll end with any questions if you have any. Also, if anyone is stuck at any step, please ask me questions, or I'm here to help, so let me know.
There is a question. If we get stuck during a real submission, is there anyone that we can contact for help?
Yes, so we do have a dedicated help desk that you can send your query to, and they will get back to you with any support or guidance that you need. I will just send a link to the support form in the chat as well. Okay, I'll just do a quick demo now since we have plenty of time. I will start from the study. So initially when registering our study, we can see here I'm logged into the web and portal, the test server right here. We often also have cases where users submit their data to us just fine and it disappears, and that's because they've accidentally submitted it to the test server. So it's always important to make sure that you know which server you are on, whether you're testing your data, you should be on the test server, or whether you're actually trying to submit your data. Because the accessions that you receive do get recycled. So because the test server accessions don't last, they get recycled to the production server. So if anyone else has submitted their data, they get that accession, and then there's a bit of confusion there just to make sure you know what server you're on. Right, so I'm on the test portal service. And you can see right here, you have the metadata model, nice and clear. And then we're going to start with our study. So I will register my study right here. I will give it a name. So I will call it mouse genome study. And then the title. Oh, there we go. And an abstract. So details about study. And then you have to give it a release date. I do not want this to be released anytime soon. I'm still working on this, so I'm going to give this the farthest release date that I can, which is exactly 2 years from today. And I can keep postponing this as much as I want, or I can come back tomorrow and I can say, okay, I want this released tomorrow, and I can do that as well. If I want to add any publications, I can do that here. I can search for any publications right here. This option right here, will you provide functional annotation? This is because any annotations that you provide to us are required to have a locus tag. So the locus tag prefix is registered with the study, and then the locus tags are included with all the genes that you submit. So you tick this if you want a locus tag prefix to be associated with this specific project. We can obviously come back and do this later on as well. Study attributes is if you have any kind of collaborations, and let's say I'm part of a project and we want all the studies submitted as part of this project to have this kind of tag that we can recognize them or search for them easily. So I could say something like, okay, study tag, and then I can name my project here so that everyone who searches for this can actually see all the projects listed under this, and I will then submit this and I will get my project accession. This project accession is very important because when you any publication you're working on, you will most often cite this or reference this within your publication. And I can access this in the studies report as well. Right, so that was my project. Moving on to samples, I will register my sample next, and we have a whole range of checklists here, and you can explore these based on what type of data you work with and what type of data you submit. If there is no checklist that is relevant to your data type and you feel like nothing really aligns with it, then we do have this ENA default sample checklist, and this works for any type of data. It is a minimal standard checklist. So if no other checklist is relevant for your data type, then you can use the ENA default checklist. Right, so in this case, we're using the Mixes host-associated checklist, and I can see these are all the fields, and these are all the helpful descriptions that I can use to see what type of data I need to input here. The recommended fields are checked by default, but you do not have to provide these. If you do have these, or if these are relevant, then you should. But in this case, because we do not have a sample that this one is derived from, we do not need these. And then you also have optional fields. If you do have any of these, please check them and make sure you provide these. And you can see this field right here actually shows you whether you can add any kind of text in this field, in which case it would say text field, or whether there are very specific values that you have to include for these fields. Right, and then we're just going to go next, and we'll download the template. Now, I already have one that I've pre-filled just to make this easier. Right, so this right here is my checklist. I've included the tax ID, the scientific name that goes with this tax ID, the sample alias, a title, a description, the collection date, the geographic location, the coordinates, and any type of metadata field that was required here. I have included. One of the very common errors that we encounter is users often erase this units field, and oops, don't think I'm sharing the right screen. Sorry about that. Right, hopefully you can see this. So often what happens is there's a hashtag units row right here, and users tend to erase this, and then they start entering their data here. and this runs into all kinds of errors. Always have to leave this intact. This just shows the units for relevant fields. And you have to enter your data below this. And if I'm registering 50 samples, I can do that within one spreadsheet. So I can enter multiple samples here, or just one, but I can also enter through one spreadsheet, I can enter up to 10,000 samples. So it's quite useful when you're dealing with a large number of data. Oops. Okay, so this is our spreadsheet, and I will just try to submit this right here. And I will most definitely run into an error because I do know what's wrong with the spreadsheet that you will see in just a minute. There we go. So the error right here is the collection date, and it shows in a very confusing way what the correct way is to include collection date. And oftentimes researchers don't have a collection date, in which case you can include values like not collected or not provided or missing, and then you have to provide a reason for why this is missing. So collection date should always be in the format of year, month, and day. And Excel tends to default to this format. So I'm just going to correct this here, and I will save this. And then if I go back to this and try and submit it now, hopefully it should go through. Right, there we go. So that is my sample accession right here. And moving on from samples, my last step would be to submit my reads. And in a similar way, these are all the read submission checklists that we have. As you can see, you have one for CRAM files, BAM files, FASTQ files. In this case, since this is not a paired FASTQ submission, this is a single FASTQ submission, I would use this checklist. And you can see there are optional fields with this as well. Not so many, but still some. And then I can download this as well. And I can then open the pre-filled one. Yeah. Right, so if I just show you my pre-filled one, you can see all the values in this checklist. So the sample accession that I want to submit this to, the project accession that I want to submit this to, the instrument model that I used for this, the library name, and this is just an alias that you're giving to your experiment, the library source, library selection, library strategy, the library layout, and the file name and its MD5. And you can see if you're confused about any of the values, you can go back here and you can actually look at their descriptions. So, oops. Right. If you go back to the web and portal and you just check this box right here, you can look at their descriptions, and this will hopefully clarify what these fields require. And I can then try and upload my filled spreadsheet. And there you go, that's my experiment accession right there, and that is my run accession. And that completes the submission of raw reads for me. So now I only have to worry about assemblies and annotation. And for that, you would have to use Web and Cloud. Okay, hopefully that was clear and everyone managed to learn something and managed to submit the data that we had in the exercises. If anyone has any questions at this point, I'm happy to take them. Otherwise, I think we can end a bit early.
I think you're off the hook there.
Great. Thank you very much. This was a great session. And I hope you guys learned a little something too. Thank you very much.
Thank you for running the session. Thanks everyone for coming, and hopefully I'll see you all in the next one.
Great.
Goodbye.